My name is Dr. Kate Wright, and welcome to the Thomas H. Gosnell School of Life Sciences at RIT. In this lab, undergraduate students will learn important molecular biology skills that they put to use in advanced coursework, advanced lab work, and also their future careers in science. In our hands-on lab course, students learn how to work with and genetically manipulate E. coli bacteria. Later on, they learn how to isolate proteins, DNA, and RNA from these laboratory strains. Here we see colonies of E. coli that have been transformed with a gene for green fluorescent protein. Students get really excited when they see their bacteria fluorescing green. In addition, students will isolate proteins from these transformed strains and use gel electrophoresis and Western blotting to identify the protein molecules. Both of these techniques are very common in research lab settings. Now let's go over to our genomics lab, where students learn even more advanced research skills.
My name is Dr. Andre Hexon. Welcome to our newly renovated genomics lab. In this lab, undergraduate students are exposed to a variety of state-of-the-art equipment, one of which is the newly designed Sanger-based genome sequencer. Over 20 years ago, this machine was used to sequence the entire human genome, which revolutionized biology. We can now answer questions related to personalized and precision medicine. Genome sequencing is done much more quickly with the advent of next-generation sequencer. Here at RIT, we have the Illumina MySeq next-generation sequencer, where undergraduate students have full access in their coursework and also through their undergraduate research experiences. There's always something new and exciting happening here in the College of Science. Hope to see you sometime soon in the lab. Bye-bye.